behalf of myself and my co-chair, Max Herrmann, I would like to welcome you to the Pandemic Response Panel of the SEX European Life Sciences Forum. We have today, other than Max Herrmann, Andrew Allen, and Juan Carlos Jaramillo, and Narender Mantena as part of our panel. And before we get too far, I'd just like to have them introduce themselves briefly. Maybe we'll go in alphabetical order and start with Andrew Allen.
Thank you, Florian. Hello, everybody. My name's Andrew Allen. I'm the CEO, president, and one of the founders of Gritstone Bio. And Gritstone Bio is a company dedicated to developing next-generation immunotherapies. We began life about 6 years ago, focused on vaccinations with neoantigens to drive strong CD8 T-cell responses against cancer neoantigens, the goal being obviously the eradication of tumor cells. And we developed a couple of technologies which turned out to have significant utility against infectious disease and various pathogens, including SARS-CoV-2. If we think about SARS-CoV-2, a pandemic that's still killing over 10,000 people a day around the world, I think we've all been surprised by the rapidity with which the spike protein has mutated. And thus, the first generation vaccines, which were very good at driving neutralizing antibodies against spike, have experienced some limitations because of this continued change in the spike glycoprotein target, such that the neutralizing potency of those vaccine-listed antibodies diminishes against several of the variants, most notably the recent Omicron variant. One solution is to keep boosting with the original version of the vaccine and push those antibody titers up as high as possible to still try and provide neutralizing coverage of the Omicron variant and whatever variant may follow. But obviously this requires multiple vaccinations, and I think we can see that many people are either unable to access the vaccines for repeated boosters or unwilling to receive repeated boosters. And ideally, we would look for a vaccine that has more durable, variant-proofed protection. And that's going to be a challenge if we focus on spike. And so at Gritstone, we've elected to include spike within our vaccines, but also include conserved epitopes or targets from other genes within the SARS-CoV-2 genome. And we can select regions of the viral genome which are highly conserved over evolution, presumably because of functional constraints. And some of these conserved regions can function as very good antigens for the immune system, specifically for CD8 T cells. And so we develop chimeric vaccines which contain whole-length spike to drive neutralizing antibodies, and these conserved T cell targets to drive broad, potentially durable T cell immunity. And in our first human data, which we presented in January of this year, indeed as planned and as expected, we're seeing the generation of strong neutralizing antibody titers, as well as de novo CD8 responses against administered non-spike epitopes from SARS-CoV-2. So more to come, obviously, as we continue developing this vaccine. And perhaps today we'll talk about the importance of T cells that I think is becoming more widely appreciated, and some of the challenges around how we think about measuring T cell responses in large clinical trials. I'm looking forward to the conversation. Thank you.
Thank you very much, Andrew. I think maybe next, Max, do you want to give a brief introduction to yourself?
So I'm co-chair with Florian, and I am head of the UK healthcare research group at Stifel, which is an investment bank. And covering this space. We obviously look forward to seeing and hearing more from the panel on the views in the vaccine area, as well as obviously therapeutics. But I'll leave it to the panel members for further introduction.
Juan Carlos.
Yes. Hello, everyone. My name is Juan Carlos Jaramillo. I'm the Chief Medical Officer here at Valneva. I joined the company back in October of 2020. And a little bit about Valneva in summary. We're a specialty vaccine company, which we focus on the development and commercialization of prophylactic vaccines for infectious diseases with significant unmet medical need. I look forward for the discussion in the panel and hope we can contribute accordingly.
Narender.
Thank you, Florian. My name is Narender Mantena. I look after our injectable business out of the US and Europe, as well as the global strategy. I work for Biologically Limited, which is one of the oldest and largest vaccine companies out of India. We make pediatric vaccines. On the normal non-COVID vaccines, we sell about over 500 million doses of vaccines to nearly 120 countries across the world. As the pandemic set in, we tried to leverage our capability in making large volumes and affordable vaccines that fit our technology platform. So we developed a RBD-based subunit vaccine that we recently licensed in India. And just today, we received approval for 12 to 18 as well. In addition to that, we also partnered with Janssen to manufacture their adenovirus vector vaccine. And while the vaccines have been very useful, we still find a lot of the world still not immunized with even the first dose. So we're hoping to make over 1 billion doses of vaccine to help immunize the rest of the world. And I look forward to this discussion.
And I'm very pleased to see that in our panel we span from the experimental to the very experienced large manufacturer that produces actually an enormous amount of doses to prevent various infectious diseases across the world. A few words about myself. I'm a physician and a microbiologist. I worked for the longest time for Merck, and I've started to make myself independent about 12, 13 years ago, and have been an advisor to a number of companies, particularly focused on vaccines. And for full disclosure, I have been an advisor to companies that have developed and produce COVID vaccines, including Moderna and Biologically, sitting on both of their scientific advisory boards. So I think we'll get started on the first question, which Max, do you want to take over? Because I'm going to answer the last one.
Yeah, sure. So I guess the first topic we'd like to address is sort of what next for the pandemic, and are we actually reaching an endemic phase of the disease? And it's kind of interesting. Certainly in the UK, they're talking about removing any restrictions and limiting testing going forward. So it would be interested to know, and perhaps maybe start with Andrew, what your thoughts are about the role of vaccines. I think you alluded to it in your introductory comment about the role of vaccines going forward. Now we're moving perhaps into a slightly different phase of the disease.
We surely are. I think, first of all, I do not have a crystal ball, and I think demonstrably no one does in terms of what will happen next. We have continued to be surprised, I think, by SARS-CoV-2. It is a coronavirus. Obviously, we have a lot of experience as humans with coronavirus, as we all know. And there are some interesting data suggesting that your prior experience to various coronaviruses may dramatically impact your response to SARS-CoV-2. There's a very provocative paper that came out from Oxford suggesting that when healthcare workers in the UK were studied closely during the first months of the pandemic, so the first half of 2020, there were a group of healthcare workers that never exhibited any evidence of disease, nor even evidence of seroconversion. They did not become antibody positive. There was really no evidence that they had been infected, although, of course, they were pretty much swimming in SARS-CoV-2 on a daily basis. And the hypothesis was raised, suggesting that their T-cell memory against shared coronavirus epitopes may be responsible for this apparent functional immunity to SARS-CoV-2. And that was indeed confirmed by the T-cell analysis performed. It's a small number of subjects, so this is a hypothesis generating study, but in particular, T-cell immunity to a conserved polymerase NSP12, non-structural protein 12, appeared to be associated with this state of functional immunity. So that was an interesting and provocative observation, and obviously in part perhaps goes to explain why we see such a heterogeneous response in our fellow humans to this virus. Some people, of course, get sick, and sadly, too many have died. Some people are essentially asymptomatic. And of course, our prior immunological history probably accounts for that. Well, our history has all evolved over the last 2 years, and obviously Omicron in particular, with its very high transmissibility, I think has changed the population immunity. Of course, vaccines have as well, varying from country to country. And so I think it's a little hard to predict what will happen going forward. I do think we're likely to see continued variation in spike. We've clearly seen that to this point. Whether there is a new variant that is both highly transmissible, as Omicron was, but also perhaps yet more pathogenic, more virulent, we obviously hope not, but that is the concern. And so I think we have to be on the alert for that and should not be complacent. I think we've had waves of complacency before, and that is a mistake that is potentially avoidable. So thinking about therapeutics and thinking about superior vaccination strategies, I think is what we should be all focused on right now. And obviously you can think about using existing vaccines and either more of the same version, newer versions of those with multiple RBDs, for example, or bringing in T-cell immunity, as I mentioned earlier, are all, I think, legitimate strategies for vaccination. And obviously there are lots of therapeutic approaches, primarily antibodies and small molecule viral enzyme inhibitors, which others on this panel are better qualified to speak to than I. But I think we should be ready for continued flares from this pandemic. And of course, let's remember, endemic does not mean safe or benign. You can still die from endemic diseases. Smallpox was endemic for many, many years. People died of it. So we should not be lulled into, again, this false sense of security, I think, by the use of the rather soothing term endemic that sometimes I think is misinterpreted to mean harmless.
Maybe quick follow up on that in terms of, do you think that we'll end up with an annual vaccination then, or maybe in combination with the flu vaccine?
I hope we can do better. I think we've learned that the annual vaccination chasing the serotype of the virus is not a highly effective strategy. It's better than nothing, clearly. But I think our aspiration is to do better and to get ahead of the virus so that we don't require annual vaccines to try and play catch up with a virus that's mutating ahead of us, but to actually develop more broadly neutralizing vaccines, perhaps by leveraging both the humoral and cell-mediated immunity side of the house, as I mentioned earlier. That would be our goal, I think.
I just want to add one comment to this, and I think, well, I fully appreciate what you say, Andrew. I think we also see that the current vaccines that are based on an original strain that is pretty far away, say, from Omicron, have preserved to a large extent the ability to prevent hospitalizations and severe disease. And so obviously there is some memory inbuilt in those. They are actually pretty close to what we would expect of a universal vaccine. The boosting problem is a big one. That's one that certainly needs to be addressed. But I don't want to leave you with the impression that we really need to adjust the strategy continuously. We don't know that yet. I mean, obviously we're trying to figure out what's best, but we're not so sure yet at this point in time. So maybe we should, in the following presentations, also talk a little bit about variants and what would prompt us to move to other strains as the predominant vaccines.
And maybe I'll ask this on Juan Carlos, the same question to you. Obviously, Valneva's approach is very much a whole viral inactivated virus approach, a more classical vaccine approach. How do you view the question in terms of what next? Where are we in the pandemic move to endemic, and then what do you see for going forward in your crystal ball for vaccinations? I think you're on mute.
It's true. Right now, no one has the crystal ball and can actually anticipate what is coming. I think the most important part where I think certain points have been highlighted already is that we know that as the virus will continue to circulate among the population, of course, there's a significant risk for further outbreaks. I think what we need to further have data on and the vaccines that are currently on the market and the ones that are hopefully coming for approval. For example, VLA 2001 are COVID-19 vaccine is that we can also show from a durability of the immune response will also be an important factor to determine the optimal regimen. I think yet having the position of that if this will become an annual vaccine like flu, I think we still need a little bit more data in order to further look into what would be the optimal either dosing schedule or the optimal variant that we need to address as the pandemic is still is ongoing and there is scientific rationale that there might be another wave coming probably after summer, autumn of this year.
One maybe follow up on that is do we know? I mean, obviously we've had first dose, second dose, booster doses now. Do we know how long the antibody titers and maybe some of the T cell responses last after 3 doses, or are we still kind of still discovering the durability of the current vaccines from a booster perspective?
So for our vaccine BLA 2001, we're generating that data as we speak, and that's also going to be, I think, an important differentiation perspective. I think for us, at least for Valneva, that is an important part of what will happen in the future, and also what other companies will probably need to strive for in obtaining the optimal either dosing regimen and so forth.
So do you think that having that additional antigen incorporated a bit like Andrew was saying, that could give you a sort of second generation vaccine that may not be so dependent on spike and so sensitive to spike mutations that render them less effective for viral mutations like the Omicron strain?
I do believe so. I think that also now and has been mentioned, T cell is an important area that we need to continue to investigate. I think as the vaccines have been more and more into the individuals, I think this is an area where we have to have more data in order to really look at how we move forward with this.
And Narender, what's your take on the future aspect for one? Are we moving from pandemic to endemic, maybe in different parts of the world? We're in different phases as well, and how do you see the vaccine? How do you expect them to be administered going forward in the long term?
No, I think you're right. I think there is a movement towards towards the endemic. It's probably forced to a bit for the pandemic fatigue, as well as success of the vaccination, along with a little bit of natural infection. I think one of the challenges going forward, well, there are 2 challenges. One is the biggest one is the persistence. I think that's something that all the vaccines have to look at. It is not practical to give so many boosters. And the second thing, even going to an annual booster, we do face one challenge that I believe that we have to address. Historically, vaccine has been associated with preventing of infection. That's been the perception. That's been what the measurement of a good vaccine has been. Now, changing that to saying that it prevents disease, I think that that also is causing a bit of vaccine hesitancy. I think that as a vaccine industry, we do have to look at that and see whether we can develop vaccines that prevent infection. I think that would lead to whether it's annual booster or every 3 years. I think that the pathway has to be towards that type of a vaccine rather than what the current vaccines, which they have done a good job, which is just preventing disease. I think they need to try to get preventing infection because managing disease is very difficult in the emerging countries when you have an outbreak like Omicron.
And do you think that people liken it to flu, but that it's more like the other coronavirus and rhinovirus and RSV type infections, where the exposure you get in early years of your life then give you protection from severe disease in later life, so that as we gain more and more experience of exposure to the SARS-CoV-2 in its various guises or variants, that we end up with it not becoming a problem at all, and therefore vaccines are not required in a repeat.
I mean, I think it's probably too early to tell. I think that we only have 2 years of data, at least with this version of the coronavirus. It appears that with the variants coming, it is the earlier vaccination is still preventing hospitalization and severe disease. So it appears that way, but I think that, as you know, we don't know what kind of variant will come in the future. So that's always a bit of an unknown. I think it's too early to do that. I think one of the things that we're doing is, while we developed the vaccine, we initially went with the 2-dose, but that was prescribed for the pandemic. We're going back to the blackboard and saying, what is the regimen to truly create a vaccine, and doing the classical dosage studies, dose interval studies, to see whether we can build a vaccine that lasts for more than 2 years, which we would normally do. So we're doing that in parallel, and naturally, and we're doing in the younger 2 to 5 population.
Okay.
And I would just like to add congratulations to Narender on the approval for 12 to 18-year-olds. I think it's a really important goal, and that's why this is so important to as Andrew also said, to basically make the whole population as far as possible experienced with the virus or the vaccine, so that they will have memory, T-cell responses, and as a consequence get boosted when they acquire the virus. And one of the good things of the research from various companies and public entities that has come out over the last period is that pretty much everything boosts in a cross-boosting way. So you can boost the different vaccine modalities with each other. There's pretty good memory responses. So I would argue that it's not just spike proteins, it's also not just non-spike proteins, it's also spike proteins that have conserved epitopes that actually generate good memory. So people who have been previously immunized react quite differently from those who have not been immunized, and generate a very quick antibody response when they encounter the virus or when they get boosted. And I think that is what makes me hopeful that we will not have to have as frequent boosters as it currently looks in some quarters. And I think on the question of whether we should protect against infection or disease, of course, I'd love to protect against infection, but that is where the problem of the variants comes in. The variants do break through much easier in terms of causing infections than in terms of causing disease. So the immunity with any of the vaccine modalities that I've seen so far wanes quicker against protection against infection than against protection against disease. And therefore, while this would be great, it may not be so easily achievable. So I think we'd all be interested in it, but we may not get there.
And Florian, maybe switching tack to the role of therapeutics, going forward as we perhaps move into an endemic phase. We've clearly got a much greater armament of therapeutics, but how do you think that they will interplay with the vaccines?
Thank you, Max. I think the development of therapeutics is obviously critical. As I think Alan mentioned, more than 10,000 people die every day. So once you get infected, the vaccines don't do much for you unless, with the exception of already having provided a basic set of memory and potentially preventing severe disease, but they don't prevent infection anymore. You're already infected. So now you do have treatment. You do need treatments, and particularly immunocompromised populations and populations that for other reasons, because they have comorbidities, cannot fence off the virus as well as we would like, or who have secondary complications, need appropriate therapeutics. And I would say there are 2 classes of therapeutics that I want to talk about briefly. One class is the class that interferes with viral replication or viral take. Those would be monoclonal antibodies. There would be antivirals as such. Fortunately, we now have at least a few antivirals. The advantage of the antivirals is clearly that they're not so dependent on the variants. In other words, you can treat, because they work on the same principle, you can treat all the variants with the same antivirals. The disadvantage of the antivirals is that the therapeutic window is relatively short. You have basically, the ideal situation would actually be to give them, and we've gone through that with Tamiflu and in flu for a long time. It's actually a very good and very effective drug against flu strains that are susceptible. But if you don't give it in time, it doesn't do much. And we have a bit of the same situation here again. We have now very potent antivirals, but you have to give them early enough, which means you have to be diagnosed early. And ideally, in terms of preventing later bad outcomes, you would like to give them as soon as you know that you're infected, or even before. In other words, as a pre-exposure prophylaxis. Now, that of course means that you have to treat a lot of people with the concomitant logistic and resource problems in order to prevent a relatively small number of hospitalizations and death. That's the biggest problem in a way. Otherwise, they're great tools. The monoclonal antibodies similarly are very good tools, and they keep being further developed. Of course, they are more susceptible to the development of variants and spike proteins. So the first generation of monoclonals are already pretty useless because they no longer neutralize the spike proteins. But there have been several new generations developed, and there's still an active development going on, so that we actually have somewhat supply constrained, but we do have some monoclonals that are active. And we can now, for example, treat people who are immunocompromised and who we know get infected, and that's the recommendation of the NIH here, for example, with monoclonal antibodies in time, and then the likelihood that they will have a severe course is highly reduced. So I think this is actually an area where we still need substantial research. We need to further develop more antivirals that are broader, that are easy to apply, and we need to think right about how to use them, which also goes along with appropriate, simple and cheap, fast diagnostics. Because if you don't use them early in the infection, their usefulness is limited. Then there is a second class of therapeutics, which we haven't really discussed here. But once the viral replication is no longer in the forefront of the disease, and it is more, I would almost call it an autoimmune disease in a way, in which the body overshoots in its immune responses and attacks itself. You need to manage that part of it, which is no longer an antiviral problem. It is much more an appropriate mixture of immune suppressive with appropriate supportive measures that then are used as therapeutics. And just to remind people, I mean, one of the most effective therapeutics in this later stages is a really old one. It's different versions of cortisone. So we have, in the meantime, learned to manage these later stages of the disease much better than in the beginning. So we lose less patients. We also deal with ventilation and positioning and physically care better. So there's a lot of things that have improved, but we still need additional resources on the latter part, the treatment of the secondary effects of COVID as well. And then finally, there is the long COVID, which is actually problematic because it is often the outcome of a very mild COVID disease or a relatively mild COVID disease in which the patient is not particularly symptomatic at first, but then has a very, very long course to recovery with severe neurological problems later on. That is, I think, an area of the disease where we do not have a good idea of how to treat it well at this point in time. So it's more than likely that antivirals would come too late, monoclonal antibodies would come too late, and I'm not so sure that the immunosuppressive regimens that we use for the secondary acute phase would be helpful either. So I don't know what we need there, but we need treatments there that are better able to deal with this. And it's going to be a pretty massive problem because the frequency is fairly high.
And that kind of raises some real issues with the testing, because I heard the UK Prime Minister yesterday comment that the UK is currently spending 2 billion sterling a month on COVID testing, and they obviously want to reduce that quite significantly. But with the symptoms of COVID now seeming pretty similar to a common cold, if you're reducing your diagnosis, then how do you use your therapeutics early on in the disease setting?
That's a real conundrum. It won't work. So, I was reminded of that when I sort of lived through the second phase of our very large placebo-controlled trials, and we kept following the same patients, and we kept seeing respiratory disease, but because the vaccines were so highly efficacious, it wasn't so much coronavirus anymore. Now we started seeing rhinovirus cases and flu cases and various other things. If we had not done the testing, they looked the same. The physicians couldn't distinguish them. If we hadn't done the genetic testing to figure out exactly what people were infected with, we wouldn't have known. And so that is a problem for antivirals, for monoclonal antibody, and pretty much for any therapy, because we, unless we have a therapy that is completely ignorant to the infecting virus, and such a thing doesn't really exist, then I don't think we can forego testing. We need, if we want to make the antivirals and the monoclonal antibody therapies useful, we have to continue to test.
I think there's some certainly things to be worked out in terms of how we position therapeutics going forward if it becomes an endemic. Maybe moving on to the lessons that we've learned from this pandemic. Maybe the most important thing out of this pandemic now we're at the stage we are is how does that position us for future pandemics? It would be horrible to think that in however many years' time we end up in a similar situation. I know it was said the world was expecting a flu pandemic, but we weren't expecting a coronavirus pandemic, and we were very ill-equipped for that. What lessons do we learn from this pandemic for the next one? Andrew, perhaps.
Yeah, I'm not a public policy person, so I'm not going to speak to that, but of course there are enormous implications for policy in terms of what we have learned and preparedness. We work with CEPI, who obviously are a trans-governmental organization that obviously is focused on epidemic and pandemic preparedness. So clearly that's, I think, part of the solution and an important part of the solution. I'm a vaccine developer, so I'll keep my comments to that side of the house. The thing that I find a little frustrating about the information we've gathered with this pandemic is our lack of insight to the importance of T cells. And it sounds very nerdy and in the weeds, but actually I think it's pretty important because we don't know what the next pandemic could be, and some will not be terribly well protected against using antibody-centric vaccines. So as an example, Ebola is a disease that obviously I'm not proposing that will be the next pandemic, but Ebola vaccines rely upon strong T cell immunity, and that may be relevant for whatever we're dealing with next. And we have not done a good job of understanding the nature of T cell responses to the vaccines. Part of that is because the assays are hard. So traditionally, to understand a T cell response, you have to collect live cells. You have to separate cells right there in the clinic, spin them, and then freeze them, and then ship them. And that obviously is hard. Most centers can't do it. And so in trials of tens of thousands of subjects, we get T cell data on tens of subjects. And I think that is a huge problem because we're seeing this dissonance with the first generation vaccines between the effects on antibodies and potentially the effects on T cells that I don't think we really understand. And of course, I agree with Florian that the first generation vaccines have been incredibly effective at controlling death from this pandemic. But we've learned a lot of things that are quite interesting with, for example, the data out of Israel, which obviously are very compelling data because of the broad population coverage with the Pfizer vaccine. We learned that after 2 doses, mortality, not infection, but mortality increased after about 5 months. And a third booster dose helped reduce that mortality. So 2 doses was not giving us complete protection by any means. Now, it's still better than being unvaccinated, but it's not as good as we perhaps should aspire to be. And the third booster dose was required there. And that suggests that perhaps the immunity is being more driven by antibodies with those mRNA vaccines, because of course antibodies wane, we know this. What's interesting about the adenovirus vector vaccines is that they historically have been used when you need to elicit a strong CD8 response. And the mortality from SARS-CoV-2 after vaccination with the Johnson & Johnson vaccine appears to be a straight line for at least 6 months. So it's starting to look a little bit different from the pattern associated with the mRNA vaccines. And if that's true, and it's a little early still, but that is the emerging pattern, then we need to understand that. And I think we have lots of hypotheses about T cells, but precious little actual data. And we're obviously now very focused on understanding the nature of T cell responses. And there are some new techniques available, adaptive in particular. It's important, I think, their notion of looking at T cell receptor clonotypes in the peripheral blood, because you can do that on a routine purple top, EDTA blood tube. You don't need any careful handling. And that perhaps is going to give us a window into the nature, quality, and quantity of T cell responses that may then correlate with outcomes. I think understanding T cells and vaccines is a huge area of important focus for us if we're going to do well against whatever pandemic comes next.
Juan Carlos, what are your thoughts on the same topic?
Yeah, so for me, I do believe is that regarding the pandemic and what lessons learned, it's really looking at some countries, for example, approach it in a broad portfolio, having both the therapeutic and also vaccines, not only just to the new technology which was coming out. I also believe that from a public health perspective, one of the things that we saw that did not work was lockdowns, for example. We saw that with the Alpha outbreak in the UK, for example, also Delta, and then Omicron. Some countries were actually shutting down travel. There was more restrictions on the individuals having the opportunity to travel to other countries and so forth. So we saw that did not work. I think we need to look at more from a perspective around surveillance and share more knowledge, more data in order to make sure to prevent this. And it goes, again, the capacity and also the capabilities to do the right sequencing for us to understand what new variants are coming out there, what can we see, and especially for other regions or countries that have probably low vaccinations, the high risk and possibility of new variants of current concerns coming out of these regions.
Narender, how do you view the lessons learned?
I think, in spite of the number of unfortunate deaths, surprisingly, I believe that we did remarkably well, given the nature of the pandemic and just the scope of immunizing the world. If you look at what we've done in the last 2 years is, yes, Africa has been left behind, but with the work that what Andrew mentioned with CEPI, as well as governments like the US, Europe, and China and India, in a span of 2 years, India has been able to immunize most of all of their adult population. China has been able to immunize all of their adult population. And I think that the industry and the governments have done remarkably well. As I said, it's unfortunate to say we have done well with so much morbidity, but if you look at what could have happened, it could have been much worse. Having said that, I think that we have learned lessons. As I said, I think while we talk about vaccine equity, it's always difficult to give everyone equally when you have different countries and different manufacturing. I think the only way to overcome that is to have more capacity. And I think that the pandemic has allowed us to develop new technologies that we perhaps you can rapidly manufacture vaccines like the mRNA, and also the classical technologies, just recombinant as well as inactivated. I think the world has much more capacity built in. And I think that countries have recognized and institutions like CEPI have recognized the necessity of funding multiple programs across the world. So I think this lesson, if we continue to remain learned and continue to invest in it, should prepare us much better for the future pandemics from just an infrastructure perspective, and how we need to look at pandemics on a global stage rather than a country stage.
Right. I mean, clearly, it's hard to. I mean, from my view, it'd be hard to beat that. I'm trying to remember. Probably, the sequence came out in January 2020, and you had the data on the first mRNA vaccine in November. I mean, it's from a phase 3 study with 30,000 patients. It's hard to see that could be accelerated significantly from there. Would you agree with that?
I would think so. I think that the technologies, not only mRNA, the adenovirus, and eventually the recombinant proteins. I think the technology we seem to have much more armaments to do it. I think now, because of the investments we made, we have the capacity to rapidly immunize within 1 year. I think that that's an amazing feature. I think that the challenge is going to be as you all know, vaccine facilities are high fixed costs. How do we maintain them? And that's going to be a challenge across the world going forward. I think, as I said, it's hard to say people have overinvested. There's no such thing when you're in a pandemic. But at the end of the day, we've been in the vaccine industry for 70 years. We know what it means of having fixed costs and lack of vaccine interest when there's no pandemic.
And you can't really mothball these sites, I take it. It's not like you can have, this is a facility that we're waiting for just in case the pandemic happens, or is that possible?
I mean, you can. I think for us, we're a vaccine company, so we do look at repurposing for our classical vaccines, and we focus on technologies that can be used for other vaccines. I suspect mRNA companies will use this in the investments in other therapeutics. So that's for a couple of them, but and I don't want to demean this. When other countries want manufacturing facilities, what they have to think about is what will they do when there is no pandemic, and it's a challenge. And we've seen governments own facilities, and we've never seen them being profitable. They're either shut down or sold back to the private industry. So it's a good thing. There's a lot of capacity and capability, but it has to be managed and nurtured well to prepare for the next pandemic.
Yeah, I would just like to echo what Narender is saying, and I thank you for Narender for bringing that up so clearly. I think we have not overinvested in vaccines in the last 2 years. We have underinvested in vaccines for decades before, and that is part of the problem. We still suffer from the perception, even among charitable institutions, that prevention has to be cheap, whereas therapy can cost almost anything. And I think in this pandemic, if we have learned nothing, we should have learned that we have saved a huge number of lives. I don't want to put an exact number on it because I don't think it's possible through the fast development of vaccines. And when I think back over my own career, development of the varicella vaccine, for example, has taken 17 years from getting into the clinic to getting it out there and licensed. And that's, now we've done this in under 2 years, which is fantastic. So we've really gained a lot. We still have major problems in the public because a large part of the public hasn't gone along, even though good tools are available, they're not willing to use it. And I do think, as Narender said, we need to sustain this. We need to actually make a model available in which it becomes not a dream, but a reality, that we finance the immunization of the world in a relatively quick time. The cost of immunizing the whole world against corona is ridiculously small compared to the costs of maintaining the consequences of a pandemic or even an endemic, because of work loss, because of all the disruptions that we see. So I think what we need to end on is essentially saying we should continue to invest in vaccines. We should continue to make them available and make them available against more diseases. And the more we work on it, the more we'll be prepared for the next pandemic. The mRNA vaccines could be developed so fast because we had been working on other coronaviruses before, among other things. The same is true for the subunit vaccines. The people who've helped develop them have also worked on other coronaviruses before. If you don't do that and don't fund it, you don't have the tools to combat a pandemic. The same is true for the T-cell responses that Andrew is talking about, by the way.
Yeah, and Florian, just to add, I think also there has been, at least from my opinion, a sense of awakening from governments. You can see BARDA now providing more funding for vaccine research. You see now in Europe here, HERA for example, compiling more and more information, more needs. So I do believe that this pandemic has taught us some lessons, but at the end also, I think, the way that everyone came together and like Narender was saying is, saving lives. We were able to manage this in a good way, and it was a good outcome at the end of the day.
Right. I think, given we're fast approaching the end of the panel, I'll probably just wrap up and thank the speakers here, panel members. Andrew, thank you very much for presenting today. Juan Carlos and Narender, and obviously Florian, you and I for hosting the panel. So thank you guys all, and we'll look forward to seeing the results of all your hard work in the vaccine area, and maybe some therapeutic advances as well going forward as well. Thank you guys so much.
Thank you.
Thank you.
Thank you.
Thank you. Bye-bye.
Bye.